Okay. Well, thank you all for joining us on the Cascade Mycological Society's YouTube channel for tonight's invited guest, Buck McAdoo. Buck's joining us from virtually from Bellingham, Washington, to talk about his book, Profiles of Northwest Fungi. Um, Buck previously described a lot of the composites of mushrooms in the book or in Buck's Mushrooms of the Month articles and Mushrumors, newsletter of the Northwest Mushrumers Association. In the book, he provides profiles of the morphology, microscopic features, ecology, global distribution, associated species, and lookalike species. And the book represents about 40 years of Buck's anecdotes and research. Buck is one of the most prolific mushroom hunters and identifiers in the Pacific Northwest. His initiation into coastal fungi began in San Francisco in the 1980, and then he moved to the Pacific Northwest, where he became a charter member of the Northwest Mushrumers Association in Bellingham. He's also a member of NAMA, the North American Mycological Association, has contributed articles to Mushroom the Journal of Wild Mushrooming, and has a mushroom named after him, Agaricus buckmcadooii, as a tribute to his contributions to agaricus science. Although his main focus has been on Northwest fungi, he has also forayed or collected in France, Scotland, England, Spain, Greece, Tibet, Colombia, Switzerland, and Belize. Wow, it's quite a list there, Buck. Welcome to the Cascade Mycological Society, and I'll leave it to you to talk about your book, Profiles of Northwest Fungi.
Well, thanks a lot, Chris, for inviting me initially, and also for the very generous introduction. I didn't realize I was quite so adept at all these different aspects of mycology, but I have indeed been working on this for a long time, and it is a composite. The featured mushrooms are composites of our Mushrooms of the Month for our local newsletter, and it was none other than Portland's own Maggie Rogers that started reading these little blips about the mushrooms and suggesting that I write for a larger audience. So unfortunately, she's passed away a lot, but she was a great inspiration to many of us in the Key Council. And I finally decided that I would take her up on it and try to put them all together in one guide. And so here they are. Now, there are some positives and some negatives about this book, because it falls between the cracks in some ways. It is actually a composite of what our club members wanted to know about. So maybe an electrician or a radio tower specialist would find something and say, hey, I want to know what this is. Can you please let me know what you find out about it? And then I go into all my notes, which I'll show you in a little while, and come up with a story for it and attribute the find to this particular character. So accordingly, there's also an index of the people in here to honor all these folks who found these mushrooms and brought them in. They usually not mentioned when a new species is announced, because they're just incidental to the situation. But to me, they're not, because they may have found the original rare mushroom that is being described. But since they're not mycologists, there's no point of listing them. So this is an attempt to remedy that in a small way, and it excites them to know that they're going to get a little bit of exposure from having found a rare slime mold or whatever they found. But it means also that it's very random. It's just the mushroom you might want to know about might not even be in here, because it's what they found, and they have not really contacted me about being in this index of the people. But while we were putting out the Mushrooms of the Month, they were quite excited about it. So it really helped the club, I think, in some ways, not in the major way, which is the teaching of Dr. Fred Rhodes, who started the club, and his teaching has been the main impetus to keep the club going forward year after year. But after that, I think the Mushrooms of the Month played a part, because anybody, even new to mushrooms, could go out and find something interesting. And indeed, one guy found, a new Ascomycete that hadn't been found in North America before. So it excites these guys, at least temporarily, to have made a little note mark in history in that sense. But we'll go into the next. I was just going to say that thanks for mentioning all these international mushrooms. I'll show a few in the next couple of slides just to get the ball rolling and look at something besides the title here. It's not going, you see. Well, there it is. Okay. So here it explains exactly what they are. But the Pleurotus fuscus farfari from Greece is the most popular edible mushroom on the island of Lesbos. And what's odd about it to me is it's a Pleurotus that seems to fruit on the ground. I couldn't find any wood underneath these at all. And evidently it's the one that the people like to eat the most. And it's kind of unprepossessing, a little brown thing. But I thought I'd show it because not everybody knows what the favorite mushroom on Lesbos is. And the next one in the middle, Rubroboletus rhodoxanthus. I need to say a special thanks to David Aurora on a lot of these. I consider David the world expert on the Boletaceae, and he invited me to go with him on a Spanish trip. And a lot of these mushrooms that were found, he knew what they were automatically. So thank you, David, for helping me with the Rubroboletus rhodoxanthus and many others that we found there. Some of these are turned into look-alike photos in the book, and I'll explain that in a bit. And some are just here just to show what's in these places. The Agroscybes, the Agroscybes from Tibet was amazing. It had the thick stature of a Boletus edulis, practically. I mean, they were big, chunky mushrooms. And these are photographed about 2 days after he found them. So they got kind of beat up in the ride in the Volkswagen bus going back and forth between temples and mushroom sites before I got to photograph them. But they were really unique, being such a large size. And I don't think it has a name yet, but I'm not sure because I don't collect Chinese and Tibetan mushroom books. Now, both the Tibet and the Colombia trip were part of Daniel Winkler's mushroom roaming tours. And this one was in a beautiful little town in Jardín, which is in the eastern Andes. And the sun would come out all morning, and then by 3 in the afternoon, it would cloud up, and then it would pour rain in the afternoon. And this is a great coffee growing region, and it gets so muddy in the rainy season that you have to take a gondola up to the coffee fields. And there's a little bar on top. It's a lot of fun to hang out in while you're waiting for the rain to subside. And right above it is this little red bird called the cock of the rock, which is sort of a parrot-like thing, but it's a beautiful bird with an awful noise like a sawzall going off. So when they coo at each other, it's not really a coo, it's a sawzall sound, very, very strident. And they love these particular trees because they live on the fruits that grow on it. And I can't remember the name of the tree. And the Tremella fusiformis over here is a big white jelly fungus that evidently has some medicinal characters, and it was found in the town of Medellin in a little park there. And we have stuck it here so we have at least one mushroom in the picture here. And below it is one of those poison frogs. They come in so many different colors, but this is a black and yellow striped one. It's quite common in Colombia. So we foraged here for a couple of days, and it was a lot of fun. We had great coffee, no matter what happened to the weather, and found some really interesting things. Okay, now these mushrooms. The one on the left is probably found in Rio Claro. It's a what do you call it? It's a center for biodiversity and all sorts of stuff. And it's a camp that you can go to and foray for a couple of days, and they feed you, and you have a little shack to live in. And it's a botanical center. And I think we found this beautiful pink lepiota in this area. And you're not supposed to remove any of the wildlife from where you find it, or from you can take it and study it, but you can't take it from one state to another. In other words, if you were in Maryland, you couldn't take it to Delaware. It has to stay in the state where it is. So of course you can't take it out of the country. That would be a real crime. So, but looking at it, the beautiful pink squamose caps are so arresting to me that I tried to smuggle one of these two out of the country by putting them in an old sock. And the sock I wore for 10 days in the jungle, and of course I couldn't fold out flat at that point. And of course, nobody really wanted to look into that sock to see what was there. So it did make it through customs, but however, it couldn't sequence very well. It had so many different problems with bacteria and things that were in the sock from sweating feet and so on, that the sequence possibility was not there anymore. So it was not a very good move to try to remove it. And the one on the right was photographed by Daniel Winkler himself of a beautiful lepiota on wood. And I don't know what kind of wood it was, but it was probably in Tayrona, which is a big national park right on the seacoast, quite a bit east of Santa Marta. So another wonderful day of foraying, a couple of days of foraying there also. Okay, so this is how the whole process begins. On the right, you see myself hovering over a little mushroom, which is blown up on the left is Amanita cinicoflava. My brother lives in Vermont, so it's a place I foray in quite a bit. And I was delighted to find this, but you just set it up and try to get a scientific shot that shows the vulva and the gills and the striations on the cap and the little umbo on the cap and all of that. So you have enough features to go on. And you make a collection, you put it in one of these little plastic bags and make sure you change bags all the time. In the old days, I didn't. As a result, you can't get a good sequence because the mushroom that was in the bag before leaves traces of itself that interferes with getting a full sequence of the next mushroom that goes into that bag. Excuse me for that. So right now, the important thing is to know is to always change bags between specimens and species. It's absolutely vital for sequencing results. Okay, after you make the collection, now you have to describe the thing, and this is really important, and I can't stress how important this is. On the right are my notes of species that I found, and I'm describing, even if I don't know the name, I'm trying to describe the whole thing in just a few basic sentences. It's not the kind of description that you would see in a new publication of a species, but it's enough to go on for trying to just identify it. So on the left here, I am trying to write down the description at like 3 in the morning in Tibet because we found out that we were allowed to eat any mushroom we wanted, but we were not allowed to study them. The park ranger had come by and heard that somebody was in the lodge we were staying in. It was actually describing mushrooms, and that's a no-no, evidently. And Daniel Winkler didn't know about it either, so it's not his fault that he organized a foray and didn't know about this. Nobody knew about it. And but I was able to get the mushrooms out of there safely. And if I worked till 3 in the morning writing the notes, then I'd be able to face the next day, which would be a day of going to temples. And then the next day after that would be more mushrooming. So that's the way it sort of worked. Anyway, I can't say it's some people these days just take a beautiful photograph, make the collection, dry it out, have a sequence, but they don't take notes. So there's no notes about how hygrophilous it was, how it dried, and what the taste was and what this odor was. Things like that are quite important to the identification further on. So you try to get down those particular things at first. And here I am right behind me is this microscope that I'm looking at over here. And these are some of the photos that are just chosen for random to show what kind of thing you might find in the microscope. It's always quite an adventure to go in there. It's like a giant treasure hunt. You try to look up somewhere where somebody else has done some microscope work on the group you're working on, so you have an idea what to look for. But even so, you don't always find all the treasures. And even 2 days ago, I couldn't find any Chilocystidia in a species I was looking at. And then yesterday, they were all over the place. I just went to another mushroom another specimen that was in the bag and pulled another one out, and there they were. So you really have to be persistent, and if you think they're there, you got to keep trying until you find them. Otherwise, you come up short. And unfortunately, I don't find all the treasures, so I acknowledge that, and I won't be describing any new species anytime soon. But I hope to help out in other ways. So basically, here I have the contents of the book. There's one thing that's missing is the key. We're now in our 3rd edition, and before you get to the mushrooms of the month, there's a 19-page key, which may or may not be helpful because it's a key to only the specimens that are in the book from the Pacific Northwest. So it doesn't have anything that's not from the Pacific Northwest in there, and it's a random key because it's just what the people found plus the lookalikes. And you'll see in a bit how the lookalikes photographs play out. But I have a nice group of acknowledgments of people that all helped with all this. And I forgot to put David Aurora on, and he's absolutely so important because he showed me so many mushrooms, even on his own property, that became lookalike photos of the mushrooms of the month. And so I'm definitely going to, if there's a 4th edition, David will be front and center on that. After the mushrooms of the month, which will just show you a few of them to see how it goes, we have the fungi less known, which is a group of 20 or so species that are either a new species that no one's found before, or others may have found them, but they haven't been described yet or haven't been introduced yet. And some anomalies that people are aware of, but they don't really know what caused them. And then comes the April Fool mushrooms. Well, as you know, couples get drift into mushroom clubs, and there's always one who's the full aficionado, and the other who's the good sport. He just goes along because her husband or the spouse loves the mushrooms, and they want to show solidarity and all that. So while he's out in the woods collecting, she might be sitting in the car listening to her favorite soap opera, bored to tears, wondering when he's going to get back so they can go out to dinner together and all that. And so this book is sort of, the April Fool mushrooms are sort of written for them. They can enjoy a good laugh and learn something about the mushrooms. There's some kind of satire about most of these April Fool mushrooms, and they can sort of come in that way and then be entertained by the April Fool mushrooms. And they're not really very professional, but they were looked forward to every year by club members. It's just that we publish one every year just because they were used to them, I think. Anyway, they're here in a little group. We might read one or two just for fun if there's time. I've already talked about the index of the people, and then of course there's an index of the fungi that are in this book. Yeah. Okay, here's an example of it's a beautiful photograph by Dr. Richard Morrison, who's a wonderful guy to have around. He's a professional mycologist, and he comes around to tweak the microscope every now and then to make sure it's working properly, so I can keep going. So hats off to Dr. Morrison, and he's a tremendous photographer. And here's one of his examples right here. And usually this is the heading for each special mushroom that we cover. So the Admiral Bleed has had a share of taxonomic adventures is in the book. The little sentence on the right about the wine red bath towels is just another sentence further on in the book that we decided to stick here to show another aspect of it. But this is generally what you see when you go from one to the next. This is the Nolenia pseudostrictia, which keyed out really well in Largent's giant monograph of the Entomolemaceae. And this photograph isn't the clearest, but we had to take it. It was raining so hard when we found that we had to pick them up, and they're very delicate. They had to treat them with kid gloves, or they break into 15 pieces. And you could take them home. We photographed them in my ex-wife's garden before they totally fell apart. But it's a beautiful mushroom worth seeing. And here's this is Sarcosphera coronara, which is quite common in the eastern Cascades, and evidently over in Idaho it's common also. But as you can see, it's on the endangered species list. And here's another mushroom of the month. And these are some comments further on in the story about the mushroom of the month that are on the right-hand side. And so, sometimes they're fun to look at also. So we just posted a few of these just to give you an idea of what might be further on in the article. And this one is a big chunky mushroom that has the same color as Clitosobe odora, and which has a wonderful smell of anise. And sometimes the French actually add anise to the flavoring when they cook it up, to extend the anise smell. And Clitosobe odora is a good edible, but no one would ever want to cook this thing. And now, evidently, to everyone's shock, or my shock at least, the two of them sequence out to the same one. So this is some kind of anomaly and twisted form of maybe even a half gastroid form of Clitosobe odora, because they come out to be the same way in DNA and sequencing, even though they don't look alike at all. And this has a disgusting odor, and the other has a delicious fragrant odor. Yeah, okay. And here it is. The whole story here is about my friend Daniel Diggerness, who did all the drawings in the book, and they're excellent drawings of mushrooms in the most part. And he used to collect chanterelles for this particular restaurant in Fairhaven, which is a part of Bellingham. And there's a whole story in here about that. And I was drinking a beer in the bar, and we heard this commotion in the kitchen. And the owner was shouting and screaming at Diggerness because some of the mushrooms in the chanterelle pile were turning bright pink. He'd forgotten to take the Crogonthus tomentosus out of the frying pan, and they turned bright pink when it cooked. And so, there was the chef threw the whole thing out, not realizing that you could actually eat these if you dried them up first. Well, here's Jack Waits found this thing up in the hills, and I didn't know how to identify it at first because it's a very rare, quite a rare mushroom. You don't find very many of them. And in the last year, a couple of people have found these out in the Olympics, and they're also over in Canada and Vancouver Island, and they're probably I'm sure they're in Oregon also. And when Jack took it home to cook it, we thought it was the greening goat's foot, which is an edible albatross. But this is not an albatross at all. It's a polyporolitus because the spores are totally different. And when we looked it up, this is how it came out. But it also turns green when touched. And when boiled in water, it turns the color of grape juice. Well, that didn't stop Jack. He ate the whole thing and says he would eat it again, and he survived it. So suddenly we found a rare edible mushroom out of just out of chance. And this is the Amanita smithiara, which is so dangerous because it mimics the edible Tricholoma murrellianum, which is our Matsutake here in the Pacific Northwest, and they look quite similar, except that this thing doesn't have any kind of an odor like the superb Matsutake does. And Matsutake has a wonderful spicy odor, like pumpkin spice, this pumpkin spice flavor that you can put on your chicken or whatever. And this has nothing like that. So you can basically separate it from odor alone. But anyway, people who've eaten it said the flavor was quite good, unfortunately. Okay, there's a beautiful shot of three little Lepiota magnospores that we found on the South Pender Island in BC. It was in Washington. This place got unfortunately clear cut in order to make room for a vineyard, which was too bad because the vineyard wasn't the right place to have a vineyard in the first place. And so we lost the forest, but not before we were able to capture these three little Lepiotas that look like dancers in a Cosmos. Really, luckily, a nice shot. And I think Elsie Valinga herself identified this as Magnospora. Okay, this one doesn't have a little capture on the right, but it has rusty spores. So for years it was called Foliota albivolata because of the rusty spores. And this actually, when it was sequenced, turned out to be a Stropharia. They just had the wrong colored spores. So it's now in Stropharia and probably will remain there until they decide to dissect that group and create new genre for that. We'll see. Right now it's right where it belongs. And it has it's well known for its huge white rope-like rhizomorphs, which you can see here. And this is funny. This was growing in an ex-burn site in the eastern Cascades, not far from Leavenworth. And I remember up the Icicle Canyon. And I remember I was trying to pull it out, and it wouldn't come out. I've never seen anything like it. I pulled and finally had to go to the car and get a trowel and dig it out. And it's a huge sort of hard earth ball, maybe almost like concrete, the size of a grapefruit came out underneath it. And it took a while to chip it away from the stem. It was a sclerotium it grows from. But we found it later in Spain where it was just growing off of a branch that had fallen down. So it doesn't always have to have a sclerotium, but most of the time it does. The Italians collect this sclerotium and they put it in flower pots and they water it in order to get more of these fruiting bodies, more of the caps coming out. I ate the caps and they had the same flavor as Boletus edulis, but they were crunchy, crisp and crunchy, not the soft texture of the Boletus edulis, but every bit as good. It was just totally different texture, but it was very similar in taste, and that's probably why the Italians don't mind just taking the sclerotium home and watering it and see what comes up. This was a little species found by Dr. Fred Rhodes himself out in Hoya Park, and which is I think on Whidbey Island, and this has been it's very rare, but it's global. It comes up all over the place, but extremely rare. And it took me a while to get around to looking at it, and then it was hard as a rock when I did. Turns out it's in the jelly fungi group, and it has basidium. It's a basidiomycete, even though I thought it looked more like an ascomycete at first. I was looking up in all the wrong places, but it's quite a story. And Dr. Bandoni in BC was the one who identified it for us and let us know that we were on the right track finally. Well, here it is. Phaeolepiota aurea. Alaskan gold, they call it, further north of here. And it does look like a giant cystoderma in a way. And it might, I've heard rumors that it might be sent to cystoderma itself at some point, but I haven't got that confirmed totally. Okay, what I'm going to do here, this is what the entire article looks like. I'm going to read some of this, about 6 minutes of it, and move on, just to give you an idea of what these stories are like. And I'm not going to read the whole thing, because you don't want to hear the whole story about this, how the spores are roughened and subfusoid and all that stuff. But I'll read the more interesting parts so you can get a sense of what these articles are like. And so I'm going to get a book now and start reading. Phaeolepiota, aka the golden bootleg, aka the golden falsefoliota, is in that elite group of fungi that can knock your socks off from 50 yards out. It's definitely a Class A brake slammer if you happen to be driving up a logging road in the Pacific Northwest in the fall, and you just hope no other cars are behind you when it happens. The richly colored golden orange caps, massive size, great floppy ring, and uniquely granular surface texture for a mushroom this size immediately rivets your attention. The fact that the species prefers disturbed ground means that you will most likely spot it when driving, biking, or hiking along a dirt road. It is usually associated with alder, vine maple, old-growth Douglas fir, and nettles, fruiting up through thick weeds on roadsides where most fungi wouldn't find clearance. The feature photo shows more mature specimens. You don't see that right here, but if I go back a page, you'll see it. There it is, the featured photo right there. Shows more mature specimens with the cap granules washed off by rain. The photo below, which is the one on the left here, shows specimens near the button stage with caps loaded with granules. In the United States and Canada, Phaeolepiota aurea are confined to the Pacific coast from Alaska to California, being more plentiful in Alaska than anywhere else. They are found throughout Europe, but extremely rare. Ongo reports them from Japan, while Singer has recorded them from parts of subtropical Asia. Daniel Winkler has found them in Colombia. Here in Whatcom County, Jack Waits finds them year after year in the same locations. One of these is about 5 miles up the Canyon Creek Road off the Mount Baker Highway, on the right side heading northeast. The other is up the Galbraith Mountain Road that starts near Gate 9 in Sudden Valley. I have found them a couple of times along a horse trail behind Lake Padden, on the side of the Hannigan Trail, and up the Coal Creek Road from out of Glacier. The fact that I even bother to report these sites shows you how uncommon it is, even in our area. Aurora writes, David Aurora, this beautiful mushroom is as distinctive as it is rare. No other large brownish spored mushroom has golden brown to pale orange caps with a granulous coating on both cap and stem. In fact, it looks like a cystoderma gone mad. Some authors suggest that if Cystoderma amianthinum could reach this size and turn a bit more orange in hue, it would look like Phaeolepiota aurea. Others think it resembles a fleshy terrestrial foliota with a mealy veil. If it is a thrill to find a false golden foliota, it may be even more of an adventure to eat one. Opinions vary on the flavor. Here they are in no particular order. Kate Mitchell, the senior editor of a field guide to mushrooms and other fungi of Britain and Europe. Edible and tasty, she says, mainly in Central Europe. Shalkwick Berenson, pleasant. Chetto, mediocre. Rinaldi and Tyndallo. Oh, gosh, all right, what happened? Oh boy, I'm sorry, I've lost the thing here. We've got to go all the way back. Oh boy. There we go. I think, yeah. The flavor is strong and disgusting. Graham, edible, but the caution of eating a small portion the first time should be observed. I like the nutty flavor, while my wife, with no ill effects, finds it not to her liking. I tend to agree with Chetto. I found that it had little flavor when sauteed in butter, but I was intrigued by the aromatic odor and sensed that there might be a better way to cook it, maybe with soy sauce and sherry in the Oriental style. But something held me back. I had heard that about 15% of the people who tried it had an allergic reaction to it. The symptoms were always of a gastrointestinal nature. Then in October of 1989, I had an opportunity to view this phenomenon firsthand. I decided to go to the Vancouver Mycological Society's Clarence Schmock foray, which was held that year in Pemberton, about a 40-minute drive north of Whistler, BC. It was a terrific foray with about 40 to 50 folks in attendance. And the last night, the chefs decided to cook up a frying pan of Phaeolepiota aurea. It was only enough for a piece per person, but even so, about 15% of the participants experienced a bout of diarrhea. It was just as Harriet Parker had warned. They were edible for some, but poisonous for others. And in the fall of 1991, Dr. Brooks Naylor and his wife Ellen, charter members of our own Northwest Mushrumers, wrote up a detailed article for our newsletter on their own adventures with the golden bootleg. Back on October 3, 1981, the Naylors had run into a big cluster along Highway 1 on the Kenai Peninsula in Alaska. They were able to identify the species from Ben Gill's The Alaska Mushroom Hunter's Guide. The author had noted, Phaeolepiota aurea has caused severe gastrointestinal upsets in some people. Care should be exercised when eating it for the first time. This very large and easily recognized mushroom is very common in Alaska's south central area and is readily collected. Pick only the large young buttons, some as big as baseballs, and leave the older specimens. The stems are tough. Wipe off the powdery covering before cooking. The Naylors heeded the advice. They ate small amounts at first, and nothing bad happened. They ate larger amounts on 3 separate occasions. On September 26, 1991, back in Whatcom County, they found Phaeolepiota aurea again while hiking along the logging road off the Sexton Road above the fish hatchery. Excuse me. On the South Fork of the Nooksack River. Excuse me again. They found 3 large specimens and fried them up in margarine for lunch. By 4 p.m., Ellen felt nauseous. By 8 p.m., she was undergoing violent stomach disturbances, both vomiting and diarrhea, which continued until 3 a.m. She lost about 7 pounds and took 2 days to get back to normal. Brooks was not as sick, but had 3 or 4 bouts of diarrhea the next day. What they couldn't understand was why they were able to eat it in Alaska, but not in Washington. Brooks speculated that there might be genetically different strains that vary in the amount of toxin produced. According to Watling, Orton, and Gregory, the species is considered to be a good edible in Europe. It is only in our side of the Atlantic that poisonings are reported. Intrigued by all the hoopla, deeply attracted by both the attractive appearance and the aromatic odor of Phaeolepiota, Jack Waits, our intrepid food editor, had to try it. Going all out, he cooked up 2 pounds of it in a stir fry with bamboo shoots, water chestnuts, sprouts, and sesame oil. The sprouts might have even been tiger lily sprouts. He found nothing wrong with the flavor and loved the firm, meaty texture. But later he was sick with vomiting and diarrhea for 20 hours. What none of us knew at the time was that Belgian mycologist Paul Heinemann had discovered hydrocyanic acid in Phaeolepiota back in 1942. According to Stivji, Andre, and Gessler, it is a metabolite also found in Marasmius aureates, Glotis eringii, and Infundibulli geotropa, all considered good edibles. But Phaeolepiota aurea has about twice as much hydrocyanic acid as Marasmius aureates, about 500 milligrams per kilogram in its raw state. After cooking, it goes down to 200 milligrams per kilogram, but it's still a high enough content for producing a poisonous syndrome in some people. Besides the hydrocyanic acid, the species also takes up cadmium, toxic heavy metal. The combination is not encouraging for the table. Despite these apparent negatives, Dr. Mike Bugh writes that hydrocyanic acid is present in such small amounts that it is not a health hazard. The toxin that causes the distress is not yet known. So something else might be going on here besides the hydrocyanic acid. Just out of curiosity, I Googled hydrocyanic acid to learn more about it. I read that hydrogen cyanide mixed with water produces hydrocyanic acid. Cassava leaves, widely consumed in tropical countries, contains this acid. This is why they're always boiled first. The burnt almond odor that some people can detect in Phaeolepiota aurea is a giveaway that hydrocyanic acid is present. Would boiling the species first get rid of the acid, or would the acid remain and the flavor disappear? I read on that hydrogen cyanide is a volatile compound that has been used as an inhalation rodenticide. The cyanide ions interfere with iron-containing respiratory enzymes, and the rats soon die. Another term for it is prussic acid. This is because it was originally isolated from a Prussian blue dye. Prussian acid is powerful stuff. It has been traditionally used as a killing agent in whaling harpoons. So there's an example of one of the stories that is in the book for one of the featured mushrooms. Normally there are some lookalikes, but nothing really look like photos that would go along with this article. Nothing quite looks like this anyway. Here's Jack Waits on the right holding up the Aurea before the meal. Anyway, I'll read one more that's a lot shorter to show you another aspect of what goes on. But this kind of information is what you get when you read these different featured mushrooms that you don't get so much in other guides. Yeah, so here's another part of this book that's interesting is that the bibliography is not one huge thing covering 50 pages at the back of the book. There's a bibliography that goes with each little featured mushroom on the back of it, so you can hone in on the bibliography you want to. You don't have to search for something enormous at the back. And the right is a typical Daniel Diggerness drawing. I think he just grew up drawing mushrooms in general when he was living in the woods on Chuckanut Drive, and I think they're wonderful. So whenever there's a large enough space, I throw one in. So now here's a really rare mushroom, and I'll probably read this story too. This is Squamanita pearsoni. It fruits on other mushrooms, transforming them to galls of their stems. It's a parasitic on Cystoderma amyanthinum. You can't even see the victimized mushroom here. Yeah, can you find that for me? They're alphabetical. Yeah, so it's on the right. You can't even see it. And it has an awful smell, like somebody had farted in a room, except really intense and worse than if you walked into the wrong bathroom at the wrong time. And that's probably the smell of the decomposing or decomposed cystoderma that it has been feeding on. There are also different kinds of spores that come up from that. I think I described them in the article. If I read the article, you'll learn more about it. Oh, okay. Oh, you're right. Okay. Ellen Batchelor, a member of our Northwest Mushroomer Association, up from Cedro Woolley, where she lives, had just arrived with her basket of fungi for the 1997 fall show. She had had a really good day collecting at the Rockport State Park. Her basket was overflowing with Ramarias and Russulas and all the colors of fall. Nestled among them, however, was one little Cespitose cluster of a species that made my jaw drop. It had recurved purple scales on cap and stem and a yellow stem base. I'd never seen anything like it. As Helen later related to Margaret Dilley, he removed that mushroom from circulation as if it were the Hope Diamond. The caps of these specimens were 1.5 to 2 centimeters wide, broadly conical and slightly undulating. They were squamose from the distal margin, the scales becoming sparser and smaller towards the margin. The purple-black scales were upturned at the disc, then flattened out on a pallid grayish-tan ground towards the margin. The gills were adnexed and marginated, whitish, subdistance, and subventricose. One specimen was intervenose. Gill edges were entire, wavy, and there were 2 tiers of lamellulae. Stems were 5 to 5.5 centimeters long and 5 to 5.5 millimeters thick. They were more or less napiform, narrowing at base and apex. Flattened purple-black scales at midstipe contrasted with the grayish mauve ground color that became dingy ochre yellow at the base. The apex above the scaly zone was smooth and slightly punctate. The odor was powerful and unpleasant, like past gas. The spores were white and turned dextrinoid in Meltzer solution. All species of Squamanita are rare. What defines the genus is the ability to parasitize other mushrooms. The presence of asexual chlamydospores in the former host fungus at the stem base, and the presence of both hyaline and colored spores in the same fruiting body. The first Squamanita ever recorded was called Lepiota odorata, or odorata, in 1918. In 1965, Cornelis Boss introduced Squamanita pearsoni from Inverness, Scotland. The collection had been found in moss under Scotch pine. Since then, other collections have been made in Strathy and in Wales. It has also been recorded from Mount Fuji in Japan, where it was found fruiting next to Cystoderma amyanthinum. Dr. Boss found another collection in Holland. Breitenbach and Kraslin found it in Switzerland. There's another report of a finding in the Spey Valley in Britain in a remnant Caledonian forest, and Dr. Daniel Stuntz found it at Tunnel Creek in Washington in 1975. According to Gareth Griffith, who wrote a monograph on the group, only 10 collections of Squamanita pearsonii have been found worldwide. species of squamanita fruit and other mushrooms, transforming them into galls at their bases. The gall was not collected by Helen, probably because it had already decomposed. Squamanita pearsonii, named after London mycologist A. Pearson, was thought to parasitize Cystoderma amianthinum. The chlamydospores belong to the former Cystoderma, not the squamanita. The genus Squamanita is also unique because it harbors species that have amyloid spores, inamyloid spores, and dextrinoid spores. Dr. Watling termed the spores of Squamanita pearsonii to be pseudo-amyloid, thereby adding yet another reaction. The combination of white gills, dextrinoid spores, and strongly contrasting purple-black scales separates Squamanita pearsonii from other members of the genus. Thanks go out to Dr. Scott Redhead for mailing me the breakthrough monograph on Squamanita. Without this key, I wouldn't have identified the species. So, anybody can go out and find this incredibly rare mushroom, and it's almost worth going out weekly to try to find something this exciting and this rare.
And how many times it's been found?
10 times, yeah. Yeah, it's only been, by now it's been found a few more times, but it's an extremely rare fungus. And here's the latter part of the article with the bibliography and the very weird Dan Diggerness drawing below it. All right. Okay, well, comes the fungi less known group, and I won't dwell here too long, but it's interesting to see a few of the more interesting things that we found. Here I am scratching my head over one that just doesn't seem to make sense to me. And this happened more, some of these or most of these you're going to see are in that category. Yeah. Okay. Well, every time I collected an amanita, I would send it to Dr. Rod Tellis, the world amanita expert. And so he has a collection of the amanita on the left. I don't know if he's had it sequenced yet because he's got so many amanitas to sequence, you never know when his turn will come up. But this is a beautiful amanita found in the Soul Duck Hot Springs in the Olympic Peninsula, with a beautiful dark chestnut colored cap. The stem was 29.5 centimeters long. The upper half in a wonderful orange brown squamose chevron pattern. So, and there was some elk dung nearby, and then we were just lucky that the elk hadn't eaten this on the way through. So here's an example of one of the a specimen that really might be a species novel. On the right, I call it the 100-year belief because I don't think everyone has seen this thing before. It's a brilliant belief. It's very small, caps only up to 3 centimeters wide. A brilliant orange salmon pore surface that bruised instantly. All parts of the mushroom instantly bruised dark blue when scratched. Pale yellow stem, no reticulation, and a volutinous cap or velvety cap that's very dark brown. And I only found it once. Dr. Thiers received a couple of specimens from this collection. He wrote back that he believed it was a new species, and so I dried it in the oven with the oven door open. He could not be sure I had scoured the tissue sizes, but if I found it again and dried it in the dehydrator, he would name it after me. He never even heard of me before. He has now passed away, and I haven't found anyone else who has seen it since. This entire collection was gifted to the herbarium at the University of Washington, Seattle. So anybody interested in pursuing this further is certainly welcome to go down there and try to find it. Well, here too, the last two, there's about 20 of these all together, but these are the last two I'm going to cover here from the fungi less known group. And this is one of the more popular features of the book from those who have read it and gotten back to me. They like seeing these odd things that aren't in any other guide. There are actually 19 photographs of species that can't be found in any other guide, and probably 12 or 11 of those are right here in this fungi less known group. One on the right with the dark blue Ganoderma had me really excited that I found a new species of Ganoderma with a naturally blue cap, but it's been attacked by a penicillium or something, some sort of bacteria to turn it that color. As you look at the knob right at the top of it, it's the usual dark maroon color that Ganoderma oregonense has. So my speculation is this is oregonense, Ganoderma oregonense, having been attacked by a bacteria of some sort that turned it blue. It's quite a shock to see something this large, maybe 8 or 9, 10 centimeters across with this color. Okay, and then over here on the left is the Gymnopolis with a lubricus cap. And you don't find too many of them. You usually have scaly caps. So this was sequenced, and a sequencing result is written right here in front of you. And it didn't make sense. It showed that Gymnopolis suberis of Europe was the most likely candidate of what this is. And I looked it up, and it has a very dry matte cap, not at all lubricus, with cap scales on it. and look nothing like the thing featured here. So this is another possibility of a species novel. I do have a collection of this for people who might want to look at it at some point. It's a beautiful thing. It's really, really, almost vermilion colored gills. They're really bright orangey gills. And the bitter taste, of course, puts it in Gymnopolis. So here we go with the April Fool mushrooms. Now, I'm almost embarrassed that these are in here because they aren't really professional, but they do sometimes parody one aspect of mushrooming or another. And in that sense, they're a little bit educational, if not too much anyway. We thought we'd show a guy with large teeth and a strange grin as we entered this particular group here. Okay, I'll read a couple of only two of these because they're kind of neat, but the members in our club look forward to the April Fool mushroom each year, so I had to kind of scramble to come up with one. And here we have a pancake coming off a log that looked like a new Ganoderma. So I'll just read it to show you what one of these is like, or maybe two of them because they're kind of fun. But I want to go down a little further. Oh, yeah, okay, here we go. Although new names in Ganoderma seem to appear in the literature every decade or so, none is so refreshingly novel as Ganoderma immodum, a rare polypore that refers maple to conifers in the Pacific Northwest. When Ivan Norelsky, a semi-obscure authority in the Lectiaceae, first spotted it outside his home near Mount Index, he'd sensed he'd found something unique. As he observed the fungus from his bathroom window, he noticed that the squirrels kept carrying off pieces of it into the upper boughs. It wasn't that the squirrels were carting them off that impressed him. It was the ease with which they tore off the chunks of the polypore from the host tree. Norelsky had rushed to the base of the maple. Here he discovered bits of the shredded fungus littering the forest floor. He picked one up. It was very soft and somewhat spongy with a warm farinaceous odor, most unusual for a polypore. He wasted no time. Within an hour and a half, he had it fully described under the name Spongipellis farinaceous norelsky. Then he stuck it in his freezer for safekeeping. There seemed to be no rush to publish. Since the squirrels had eaten all the other material in the area, there was no threat that some visiting polypores would show up and describe it ahead of him. He went over and started a fire in his hearth. Then he uncorked a bottle of buzzard's breath ale to celebrate. Norelsky was a bachelor and conservative by nature. Drinking beer with exotic labels was the only wild thing he did. The importance of his discovery grew in his mind daily. He would remove a specimen from the freezer every third day and check it for any new characters. He had noted originally that the specimen measured 17 centimeters in width. It had a cinnamon-colored crustose cap. It was sessile, annual, and had a thick cream-colored band around the margin, reminiscent of foamy toxins. The pore surface was more yellowish buff. The pores were round and widely separated without any particular pattern. Norelsky was intrigued by two characters in particular. One was that some of the pores could be found on the cap surface, but this means that Spongipellis was evolutionizing. The other was that the entire fruiting body was waxing paler over time. Can species of Spongipellis be hydrophanous? He dutifully jotted it down, not realizing for quite some time that a layer of ice had slowly encompassed the specimen. Eventually, word of his find leaked out. It was probably when he tried to publish it in Mushroom the Journal. There had been a brief series of board meetings when the publishers and editors had convened to discuss expansion in this direction, a flurry of activity that went nowhere except for one significant side effect. It attracted the attention of Pengus Wanamaker, the vulture of modern taxonomy. Pengus was the one personage you did not want sniffing around your genus. Although he was an accredited mycologist, he couldn't bear to describe anything new. He lived instead to alter those species names described by others. He was a tinkerer who planned raids on the genre of other mycologists with the same grim determination that one corporation would use in the attempted takeover of another. For this purpose, he always had around him a string of graduate students. There were 15 to 20 of these amateur investigators at any one time who could be dispatched around the globe looking for mistakes. When the corrected names were published, Pengus Wanamaker always included his assistants in the authorship as et al. If they hungered for a little more recognition, Pengus would wag a finger to remind them they hadn't gotten that degree yet, had they? So, of course, it wasn't Dr. Wanamaker's vehicle that broke down in Ivan Norelsky's driveway one frosty December afternoon. It belonged to an et al. and a clever one to boot. She had already discovered that Norelsky never sent dried herbarium specimens by regular mail to a university. He only sent freeze-dried material by express mail. This meant to her that the new species was lurking in the freezer. She hoped the freezer would be somewhere near an exit door. She arrived at the front door, coughing badly and pointing to her Mazda, where a smoke bomb had just been set off under the hood. Norelsky had barely time to register that the young lady was attractive. He took one look at the growing sulfurous cloud and suggested a jump. He told her he just happened to have cables in his root cellar. Now, it might not be the battery, he informed his visitor, but there's no harm in trying. He groped his way to the root cellar. His visitor didn't remain mystified for long. He scampered around until she found the kitchen. It was a miracle. The new spongy pellis was right in the front in the freezer compartment. She was out of the door and resubmerged in the cloud of smoke before Ivan returned with the cables. The specimen was still rock hard when it reached Dr. Wanamaker's mobile mycology lab an hour later. Pengus was impressed with his grad student. "You are inflammable," he cooed, "even imperturbable. And since it hasn't been published yet, we can dispense with the ex-Norelsky." He changed the genus to Ganoderma because of the hardness of the pileus. He then fished out his pocket English-Latin dictionary from his glove compartment. He sighed. He noted that Emotus was the name for imperturbable, and thus the species was named. Okay, yeah, right. Hold on a sec here. Okay, so this is the last one I'll read. It's kind of funny. About Climacodon edificialis by Smog and Hednow. And this is one that some people really enjoyed, so I'll read it really quickly. Anyone heading from a hardware store with a 2x4 cannot help but notice the soaring price of lumber nowadays. Despite the well-publicized efforts of environmental agencies, the vast amount of clear cutting throughout the Northwest during the past decade, combined with the equally voluminous exporting of timber to foreign nations, has created a serious shortage in wood. As more builders have moved to particle board and vinyl siding in response to the shortfall, it was only a matter of time before the polypores reacted. They too, like builders, have had to adapt to new substrates. None, however, has been more successfully locally than the ubiquitous polypore Climacodon edificialis. Like its closest relative, Climacodon septentrionalis, it fruits on wood, its massive clusters arising from a point of attachment only 2 to 4 centimeters in width at the base. But unlike Climacodon septentrionalis, which fruits high up in living trees, the adaptable Climacodon edificialis is just as happy on vinyl siding, blacktop roofing, particle board, and all grades of treated plywood, often engulfing 2 or more substrates in one fruiting. Smog and Hednow, 2 graduate students in mycology out of Enumclaw, were the first to spot it. They were shocked one afternoon in late October of 1996 to find a giant cinnamon-colored polypore sprouting from the roof of their local IGA food store. Smog borrowed a ladder and went up. From the ground, it looked like some kind of aborted sporasis, but on closer inspection, the lanky, saurian-looking student began to realize that no amount of wine could save this one. It was nothing but a tough, inedible polypore whose shelving fruiting bodies had fused in age to form a quirky, amorphous blob. Hednow had now scrambled up behind him, a small, excitable sort of person. He circled the specimen several times, his head bobbing like a dachshund's. What genus is this?" he barked so stridently that Smog broke out of his private funk at not finding a meal on the roof, presently offered Climacodon. After all, we had had to climb a ladder to get there. Not to be outdone by the competitive Smog, Hednow pointed out the edifice it was fruiting on. The name for the species was born. Since then, Climacodon edificialis has been the bane of builders and contractors throughout the Northwest. Here in this photo taken by Lee Whitford in Nooksack, you can see the polypore fruiting from vinyl siding while it prepares to consume the plywood housing of the homeowner's power meter. Lee had interviewed the deeply disturbed owner. "I went with vinyl to keep out the rain," he had muttered. "No one warned me about the toadstools." "Maybe it was attracted by the power meter," Lee had offered in the Applebay. "Nah, it's just nasty stuff," spat the homeowner, who preferred to remain anonymous. "I was contemplating a class action suit, but who the hell do you sue? The electric company? The vinyl people? I can't even remember where I got that plywood. The creepy fungus eats everything." Lee shook her head in commiseration. She considered inviting the man to join Northwest Mushrumors, but then realized the timing might not be quite right. She left him muttering about setting up a hotline on the internet and then drove home to develop her photos. Climacodon edificialis is a bit strange even for a polypore. The fruiting bodies are massive from the start, erupting overnight into compact round clusters. Triliocercus is crustose to tomentose, dark chestnut to cinnamon, but often paling where the margins meet wood, vinyl, or soil. While the taste is infinitely forgettable, the odor is not. Species smells like whatever substrate is fruiting on, whether tar with blacktop, pulp mill with plywood, polyurethane with vinyl, and so on. As I pored over the microscopic details in Smog and Hebnau's publication, I wondered how such a monstrosity was created. How come there were no recordings of the species from the 19th century? Why could it not be found in any North American or European mushroom guide? How had it come into being? Under observations, Hebnau suggested that buildings just weren't the same back in the day. It took a little time, but the answer when it came did not derive from the monographs on polypores. It came from the lumber yard at Sash and Door hardware store. I'd been browsing around in search of pier blocks when I ran into a discarded pile of plywood. The layers were beginning to separate. I was wondering if humidity alone could have caused this problem when I noticed the mycelium. Stringy, pale orange rivulets of mycelium were permeating the plywood in all directions. Sorry, but that's not for sale, a yard hand had hurried up to explain. Perhaps he had misinterpreted the intensity of my interest. Terrible experiment, he went on. This company, I just can't be more specific without facing a libel charge, had invented a new glue for the ply. I'm sure you've heard how glue manufacturers have studied how limpets adhere to rocks in ocean storms to formulate their glues. Well, some genius thought, why not do this with shelf mushrooms? Some of those bracket fungi are stuck so hard to tree trunks you have to saw them off. He then winced and gestured towards the plywood stack. I followed the sweep of his arm, and there at the very center of the stack was a 3-foot diameter fruiting of Climacodon edificialis. And so that ends the two of maybe 10 or so or 12 or so false mushrooms of the month. But some of our members enjoy reading them, so I thought I'd read a couple of them to you. And now I'll get to a more serious part of the whole thing. People wondered how I had found so much interesting information for each particular featured mushroom. Here's an example of a page that I would copy out in either in the UW, in the Nature Sciences Library in the UW. I would get whole stacks of maybe Sesca mycologia or Carstenia or Persunia or Northwest Science or Madrono. Excuse me again. And sit down and write out all the names of the mushrooms that had some kind of descriptive value in them, starting from 1900 on. And then I would, like here's one by Albert Slip and Walter Snell, and my little symbol for it is SLI, the first 3 letters in Slip's last name. So under this, I would write down a list of all the mushrooms that were described in that particular article. And this sort of thing I did also at the New York Botanical Library and the Geneva, Switzerland Botanical Library, and even a week or so in Kew Gardens in London. And this was to educate myself on where the information would be in case we ever found these mushrooms. And I did it for temperate countries, not including China and Russia, but everything in Europe and North America. And this took up quite a long time. I think I got up to 1992 before I finally gave up the ghost. So there's an awful lot that's not covered in the bibliographies. Okay, so now we're going over to the index. So under Albatrellus, we have all these different names that are in Albatrellus, and some of them are still ongoing, and those are the ones written in ink on top, where we found each one of these little letters is a symbol of a source where you can find some kind of descriptive value for the mushroom. So under let's see, we can go to anything. Under Albogymnopolis nanus, it's in red. Red stands for Mushrooms of the Redwood Coast by Noel Siegel and Christian Schwartz. So if you go there, you'll find something about Albogymnopolis nanus. It's probably a non-prob, but there it is, and that's how this works. So this is a really important part of how I got the book together. Down at the bottom is Albugo candida, and it's in the temperate mushrooms of temperate fungi of Europe, part 2. Okay, so now here comes the bibliography itself. So all those little synonyms for, or whatever you call them, that symbolize the story, like SLI for slip, are here in an alphabetical order. And so if I wanted to find out where that particular mushroom was, and it said Howie 2, it would be the Garicales of the Hawaiian Islands, part 2, and that's where I would find a description of the mushroom that I was interested in looking at. So this is something that I've tried to put that I've actually put into my hard drive in my computer. So I hope to have it for anybody to work with if they're interested down the line. And so here's the Mega Mushroom album. It probably weighs, I don't know, I haven't actually weighed it. It may weigh up to 50 pounds or 40 pounds or something like that. But it's all the mushrooms that I think I've been able to identify, a lot with help from other experts that I just posted them in, and the bottom, there's a name in pencil on the bottom in case the name changes, which they often do due to sequencing. I can erase it and put in the new name. And on the left is the place where it was found, and on the right would be where I can find a photo, or a photo where the negative for the photograph is, and also maybe if not alone that, but also where the description can be found. Because often I typed out descriptions in the old days, and now I'm just typing them out in the computer when they come up. So here's an example of just 2 of the photos in the album close up. So if I want to find the photo for Aboleptonia sericella on the left, I would look up IMG 93, which would be in one of my flash drives. And the Varludia on the right, the description for it would be on page 39, number 3. And that would be typed out in one of my notebooks. No, I'm sorry. The 39, number 3 is the name of the negative. I wanted to find the negative of this to make another picture. 39, number 3 would be the negative, and page 36, number 3 would be where you'd find the full description of it. Okay, and this is where you can order from. This is not a mega publishing company. This GLDesignPub.com is a small mom and pop publishing operation in Boulder, Colorado, and they luckily published my book. We're at the 3rd edition right now. There may or may not be a 4th one. We'll see how it goes. But if you want to order it from the company, the www at the bottom is where to go. This company is unusual because they also their main attention is not on publishing fungal books and that sort of thing, but on their charity, which is called MAP. That stands for Musical Ambassadors for Peace. And they have a theory that music can heal the world of all its ills. And they proved it over and over again. The founder of the company, my great friend Cameron Powers, actually flew into Kuwait a day or 2 before George Bush began to drop the bombs on Baghdad. And he and his girlfriend, who plays the drums or some other instrument, and he plays the oud, and he learned to play it in the Arabic style. And they hitchhiked up from Kuwait all the way to Baghdad and arrived probably at midday, set up on a creek corner and started playing music that was copied from Iraqi musicians from ages back, famous folk songs by famous old Iraqi folk songs from long ago. And even though they look like Americans, she's Greek actually, but he's a redheaded guy about 6'10". It can't be anything else but an American or Canadian or whatever. They were invited into homes and fed and taken care of, even while bombs from their same people were dropping on the city of Baghdad. So from that, he got enough acclaim from doing that. It was a very brave thing to do. And he was invited to play in front of Cairo at the Great Cairo Auditorium with 60,000 people listening to it. And he was invited, he and his girlfriend, Christina, were invited to go there and play there. So I'm a firm believer of this, and I've decided to let all the proceeds from sales of this book go to this charity that they run also, and you should take a look at it. Musical Ambassadors for Peace. It's fantastic what they're doing. They're even helping people in refugee camps in Uganda. So it's starting to stretch out and has a far long reach. It's almost global by now. I highly recommend it. Go and visit that site. Okay, and this is about it, folks. this is one of the pictures that's in the book. I painted it. It's of a mycologist with his collecting basket, but he's, instead of being outside in the woods, he's actually walking along a hyphal highway, a mixed hyphae going by, and he's probably wondering how the hell he got there and how can he get out of there. Behind him is a forest of cystidia of different kinds, and then brushless spores flying around the sky above. It's very, sometimes you get so intrigued by microscopy that you just can't get beyond it. And this is what happened to this particular guy here. So if you purchase a copy of the book, you also get a full page picture of this in it also. Anyway, thank you so much for listening to all this. And if you have any questions, I'm sure there'll be a way we can touch bases later. And hope you have as much fun as I did with this field. It's magnetized my entire life. Thanks so much for listening. Bye-bye.
Thank you, Buck.
Yeah.
That was amazing. We appreciate the overview of your book and your the way you came up with it, your writings, your ability to keep track of your notes and photographs, and the bibliography is just stunningly amazing. Nice work. Congratulations.
Okay, thanks a lot, Chris. Yeah, I didn't know how to go over, but that's pretty much the way it's been. And I'm on a project right now. It's much more scientific than anything I've done before. And it's the Collybioids for the Tea Council. And it's just astounding how many new Collybioids we're going to be coming up with. And I'm going to need to have it totally reviewed by some expert, because I've had to write microscopic descriptions to all these new things. And I want that to be double checked before it goes into publication. But since they named that mushroom after me, I felt that I had to do something more scientific than this book alone, which is written for amateurs mostly who are in mushroom clubs who just want to, be part of the mushroom world. And that's what this book is about. But I'm trying to go a step up the ladder with this new work, and we'll see what happens with that. But
sounds exciting.
But I appreciate you singling me out for letting me at least talk about this book, which is, kind of controversial in some ways. So anyway.
Okay.
Not everybody's into it. Some people love it and some people just can't stand it. So it's controversial in that way, I have to tell you.
Well, I loved your presentation.
Okay, thanks a lot. Great.
Annie, do we have any questions for Buck?
Well, I have a few questions first.
Wonderful. Okay.
So this is the 3rd edition, because I can also find this title on Amazon and some other, like Barnes and Noble. So I was just curious, are those 1st and 2nd editions, or?
Well, the 1st edition didn't last very long. I think maybe it was so crude that we just maybe published 3 or 4 books out of it. So that was, but we had to name the 2nd edition in order to get it out right.
So did the
2nd edition? Yeah, go ahead. Sorry.
Did the 1st and 2nd edition have the same book the front cover, or is that only on the 3rd edition?
No, they had the same cover. The 2nd edition had the same cover. The difference was that I hadn't sequenced some of my things, and one of my featured mushrooms was a Satharella, and I couldn't find any velar material in it, so I keyed it out wrong. And turns out that some Satharellas lose their velar material as they get older. So I was like 60 pages off in the key in the Satharella monograph by A.H. Smith. So I had the species wrong. And then it turned out that not only that, but it looked like it could be a new species that I had featured. The sequencing showed it to be potentially a species knob or 2 or 3 other things. So I said the best thing to do is just remove it. So I replaced it with Gymnopolis luteofolius, which is a much more beautiful mushroom and more stunning and more interesting in some ways. So that's where we went to the 3rd edition to get rid of the mistake in the 2nd edition. And there were a few of the lookalike photos that weren't quite right in the 2nd edition. So we corrected those. We had those sequenced and made the appropriate connection. So that's another reason why we went on. And there may be a 4th edition because of all the new name changes coming up, and sequences may. We're going to sequence a bunch of stuff that Danny Miller, who's my sequence interpreter, wants to have sequenced. And it's not a witch hunt exactly, but they are species that are so interesting to him in general because they haven't been reported from the Northwest before. And they're just lookalike photos here, but he wants them to be sequenced too. So we will get around to that. The sequencing is a very slow process. Most sequences are backed up years in advance, so it's really I don't know how long that will be, but there may or may not be a fourth edition depending on how long it takes to get there. But we'll see. Yeah.
Just to clarify a little bit more what you were asking, it looks like on Amazon the third edition is the one that shows up. There are the options to buy some used copies, but the price on Amazon is definitely lower. If you go through the publisher, it supports that charity that Buck was mentioning. So it's up to you what you feel like you can do financially, but definitely more of the funds go to that cause if you buy it from that site. But it is available through other sites as well, and it says third edition on Amazon. So
yeah, right, that's true. Yeah.
I was
Yeah, pretty
also
much.
curious about how many mushrooms are profiled in the book. Do you know?
How many are profiled?
Yeah.
Okay, well, they're the featured mushrooms, and I don't know exactly. I haven't made an exact count, so I can't say, but maybe over 100. I don't know, but there are the big thing is that what I wanted to do different about this particular guide is I wanted to have the lookalike photos right next to the featured photo. So you didn't have to go to the center of the book to find them. You just turn the page and there they all were. The ones that you might confuse with the featured mushroom are all right next to it. And that's what made the book so expensive. And it can't be so expensive. It costs $42 to make one copy, according to Ingram Lightning, which makes the book. And so a bookstore would have to sell it for $84, which wouldn't happen. They wouldn't be able to sell it for that. So chances are you'll never find this in a bookstore. It's not made for commercialism. And that's the problem, because I didn't know how serious this was going to be. I'm thinking that there's so many advances in cell phones and all that they might have had an advance in lowering the price of making colored prints in a book, but they really haven't. So I miscalculated there, and that's why the price is so high. It costs a lot of money to have each single page with one or two mushroom photos on it, that sort of thing, compared to what David Aurora has. They're all the colored photos are in the middle of his book, so the price is much more reasonable because of that. Anyway, we'll see. Maybe there'll be a breakthrough in technology that'll bring the price down. This book will finally have a reasonable price for selling. But if you do pay more, it's for a good cause. Yeah.
Chris, are you still there?
I'm still here. Yes.
Okay, just making sure.
Do you have other? Did we get any questions online for Buck?
No, I'm not seeing it. Oh wait, you know what? If I scroll down, no, I'm not. I'm surprised.
I did want to mention
have over 50 to 60 people watching, so I'm surprised.
Excellent.
Okay, well that's nice. Well, thank you all people who are watching. I appreciate it very much. Thank you very much. Yep.
Well, thank you, Buck, for agreeing to present, and thank you for producing this book. I did want to mention of the featured mushrooms you talked about, the Phaeolepiota aurea, that golden lepiota. I've only ever seen it once, and it was brought down to a mushroom show here in Eugene by Dr. Dan Loma when he was visiting Washington State. And I have a feeling it was probably in your neck of the woods that he collected it.
I see. Yeah. Did you get sick from it, or
no, we did not eat it, but it did win because it was such a gorgeous specimen collected so perfectly. It did win the best of show that year, and it was a mushroom I don't think we've seen since then.
Yeah, it's fantastic. Yeah, I'm sure it probably shows up in Oregon too, because they're so close to Washington. But it's rare enough that individual collections have been cited before. So
I've only seen it once as well, and it was also at a mushroom show that someone had brought in, and I couldn't believe it because it really is so stunning, and in a kind of a subtle way, at least with the color. I mean, the size is impressive, but the texture is, I think, the best part because it feels like a fine suede. And it's just, I still think about it, and I'd love to find it, but it seems not super common. So hopefully we can all find our own.
Yeah,
Yep.
Absolutely. Yeah, be careful when you eat it, because the aroma is very attractive. You just want to get into the frying pan as fast as you can, and then you may pay a terrible price later. So we'll see.
Well, always follow those guidelines. Only eat a cooked tablespoon, and make sure you've done the correct ID. One last comment before we let you go. My lovely wife Molly wanted to give you credit for some stunning artwork that you have presented here in your throughout your book. So nice
Oh, well,
work.
thank you.
Yeah.
Yeah. Well, that's amazing. I want to credit Dan Diggerness because he's had a rough time. He grew up in Chuckanut Drive, which is south of Bellingham along the coast, and his dad ran a place called the Oyster Bar, which is a pretty famous restaurant. And since it's closed down because it's right on a cliff and it's not earthquake proof. So it sort of finished up its natural lifespan. Right now, I think a new group has bought it and they're considering bringing it back, but they have to make it earthquake proof first, which is going to be very expensive. But Dan grew up sort of running errands for his dad in the club, and he was treated kind of badly. Dad didn't think he was really the right kind of person to inherit the restaurant when he went down. So Dan spent a lot of his time in the woods drawing things, and he drew fantasy fish and he drew mushrooms. And I think his mushroom drawings are just incredible. So that's why they're in the book here, wherever there's a wide enough space. And he's a person that disappears for long periods of time into the woods, and then maybe lives in a tent somewhere and comes back and joins society again. And he's back and forth between being, totally out in the woods and with people. So
he sounds like, well, one of the mushroomers that David Aurora would describe as the outsider.
Yeah, he's definitely an outsider. He's not real comfortable with groups of people, but he knows his mushrooms really well, and he studied them for a while, just for looking at books, and he just has a real talent for just drawing mushrooms with especially with rum bottles and things like that. Right? So, yeah, so quite the character. I haven't seen him for about a year, but anyway.
Buck's working on a great new piece right now, a painting that includes some Astraeus.
Could you lean in a little bit more, Lauren?
Oh, and Buck's working on a new painting that involves Astraeus, one of the sedimentary Earth star, and that's going to be a really fun piece. So we'll have to make sure you guys get a peek at that one when it's done.
Yeah, it's I've got a couple of weeks left, but I'll post it on Facebook and Instagram. So if you follow those, you'll get to see it. It's they're tearing apart an old car, so it's kind of fun. We'll see. It's cool.
Well, thank you so much, Buck and Lauren.
We do have one more. We do have a question that just came in.
Well, amazing. That's incredible.
It says are you talking about P. aura? Do you know much about Disoderma and or Squamanita?
I think Disoderma is a new name for Squamanita. It just came up. So if there is another, there it is. Yeah. If there isn't a fourth edition, which depends on the publisher, whether you think it's worth going that way or not. Right now, I'm sure she doesn't. The sales are very sparse. It falls between the cracks. But Disoderma is evidently a new name for the genus Squamanita, except for maybe the original one, which is Squamanita odoratum from Lepiota odorata. That may still be in Squamanita, but the others have evolved. This is now a Disoderma, what we're looking at right here. So. These kind of things will be corrected in the next issue. David Aurora is supposed to do a new issue of Mushrooms Demystified, but he's extremely careful about names, and the names are changing almost every week. So I don't know if he'll ever get around to it, because the names maybe keep changing for the rest of our lifetimes and he might say it might not be worth waiting for the last name to come through, that sort of thing. We'll see. Anyway, best of luck to him, whatever.
Yeah, it does seem like the naming taxonomy is still in flux, and it's probably 10 years down the road at least till things start to settle down, I would imagine.
Yeah. Well, I just want to say that it's great fun foraging around Yahats and even further north, like Tierra del Mar. We found a new species of Crogompus there that I thought was the old Crogompus locratius, but we had it sequenced and it's a brand new species. So right where you are in Oregon, there are new species all over the place, and it's so much fun to foray there, because you can just walk on beautiful old mossy trails, and right and left are potential new species all around you. It's like being in paradise if you are a person who likes to publish new species. I mean, it's really interesting to be there. Yeah.
David also asked, have you found Squamanita?
I haven't found any other Squamanita except for this one you're looking at, the Squamanita personii, which is of course now in Disoderma, that sort of thing. But you just have to get used to the name changes. And,
Yep.
and luckily the name changes will hide any mistakes I made. I can correct them.
It's too good of a name. I will reject any other names because I think it's just the best
Yeah.
name ever.
It's a very apt name for the species with all the scromules on the cap. And they're very, very strong. And on the stem too. It's Squamanita is the perfect name for it. It's too bad we have to go to Discoderma, but it is what it is.
I'm fine with Squamanita.
Yeah, okay, that's what we're using here. We'll see. Maybe we can keep it for some reason for people like myself that like tradition over perfect accuracy. We'll see. Anyway, thanks a lot for inviting me. Really appreciate it. I think that's it, right?
Well, let's say goodbye. Goodbye, everybody.
Chris,
Yes.
Yeah.
did you have any announcements or?
Stay tuned. Next month's speaker is I've forgotten his name. Brian Strait he's from Oregon State University. He's going to be speaking about his research. He's a Freeman Rowe Scholarship winner. So tune in next month, third Wednesday of February. And
Okay.
What's he presenting on?
Yep, we look forward to it. Thank you, guys.
Yeah, okay, we look forward to that. Thank you.
All right. Goodbye, all. We'll see you next month.
Okay, thanks. Yeah.